All right. Well, thank you so much, Hugh. We are going to transition pretty quickly to introduce Patty Cogswell. Patty has been working with the EHE Foundation and has been instrumental in providing her invaluable experience to us. Patty will spend a few minutes sharing some exciting news on the EHE Biobank, and I'd like to welcome Patty Cogswell.
Okay, sorry about that. Hi, everybody. Thank you, Jenny. It's really nice to be here, and I'm really excited to be part of the EHE team. I'm going to start with my just a little bit of background, so you can understand why I've joined with the EHE team. Here we go. Okay, now, sorry, I don't know if you, I can't tell what everybody's seeing. Okay. Can you advance the slide, because I don't see a button to do that? Thank you. Okay. So I spent the majority of my career as a bench scientist at UNC Chapel Hill and Al Baldwin's lab, and we studied transcription factor NF-kappa B and its roles in various cancers. But following a job change and relocation for my husband, I was looking for a new direction and was fortunate to join the Chordoma Foundation, another rare sarcoma foundation, in 2012. And my role was to develop their biobank and increase models of the disease. It turned out we were extremely successful, going from 1 cell line when I started to more than 20 cell lines and 0 PDX models to 14. And many of these models came directly from biobank tissue. So it was a really sort of exciting idea, and we proved that it worked. But through my work at the Chordoma Foundation, I became aware of the similarities between rare cancers, making it difficult to study the disease. While the diseases are quite different, the hurdles of lack of models and tissue to advance research are not. From a desire to help other rare cancer foundations, I have now formed a company, Rare Biorepository Consulting, to help build these that are lacking for rare cancers. I'm excited to take what I learned working for Chordoma Foundation and apply it to EHE and other rare cancers, some of which are listed on this slide. My hope is that not only can we be as successful, perhaps we can streamline the process and make it even simpler. Next slide. Here's an overview of what we're intending to do. Tissue or fluid draws can be put directly into various culture conditions to attempt to make cell lines or 3D organoid models. These models will take several months to hopefully develop, but can then serve as research tools to better study the disease. As we've heard in several of the talks this morning, there's a desperate need for models to better study EHE. In addition, excess tissue will be stored long-term in our biorepository for various molecular analysis, as well as research down the road once data has been generated. We'll continue to supply tissue to researchers we currently work with, but by centrally locating an EHE biobank, we can promote research worldwide. As stewards of this precious tissue, we need to be sure we're making the best use of it. I think in the speakers earlier this morning, it's very clear that collaborative work is really how we're going to advance research. Next slide. So here's where we are. We've partnered with Cogswell Institute to establish a repository for EHE biospecimens. We now have a fully operational IRB-approved protocol to consent patients and collect associated clinical data. We're working with EHE, and we're working with Cogswell, and we will have kits on hand for specimen collection to make the process really simple on the hospital end. And our agreement was finalized in December, so we are ready to go. It's incredibly exciting to see this moving forward. Next slide. There are many ways you can help with this initiative. First, help us get the word out to patients having surgery or that are having a fluid collection, or associated clinicians that they're doing this. We're able to contribute specimens if they're interested. In my experience, the majority of patients I've worked with say they would have contributed tissue if they had known. In addition, most patients aren't aware that much of the excess tissue isn't saved, but rather discarded. We're missing an opportunity to do something positive with this excess tissue and hopefully help EHE patients in the future. You can also provide input on who we might contact or help with what your research needs are. You are the experts in the field, and your input, we'll be able to work together for more success. And finally, use the models and samples once they're available. This is our main objective, is to get the samples, but to also get them into researchers' hands as quickly as possible. Working together, we can make a difference for these patients and help find better treatments and hopefully a cure for EHE. I'll take any questions that anyone has. Thank you.